So hi everyone and welcome to our weekly seminar. The speaker today is Bernadette Stolz, who is currently a postdoc at EPFL in Switzerland, and she's going to talk about applications of topology to data from oncology.
So thank you for the kind invitation. It's great to be here today. So in my talk, I would like to highlight two recent applications of TDA to data from oncology. The first one being looking at recurrence in paediatric acute lymphoblastic leukemia. This is a collaboration with, among others, Salvador, Victor, and Helen. And we published this in PLOS Comp Bio last year. And then in the second part of the talk, I would like to showcase two methods that can encode relations in multi-species data, as it is common in biology these days, with applications to the tumor microenvironment. Okay, so acute lymphoblastic leukemia is unfortunately the most frequent pediatric cancer. It can also affect adults, but it's mainly children. And it's a type of blood cancer which develops in the bone marrow on the timescale of days and weeks, and it develops from precursor cells to lymphocytes. And the type of leukemia that we're considering here is B cell acute lymphoblastic leukemia, which affects a precursor of B cells and then leads to abnormal production of B cells, which then flood the blood of these patients. Now, the good news is that chemotherapy has drastically improved survival rates. So most children that get this disease will actually survive, but unfortunately 15 to 20% of patients still relapse. At diagnosis, there's routinely a couple of different types of data that are taken from a patient, among other things, to assess the risk of this relapse. And these data include qualitative cell morphology and genetic data, as well as quantitative biomolecular data, for example, flow cytometry, which will be the focus in this talk. So flow cytometry gives quantitative information on bone marrow status at a single cell level. And what this means is that patients have a sample of their bone marrow taken, which consists of cells. And then these cells are put through a whole system one at a time, and fluorescent markers are then being measured. And so each cell gives rise to one data point, and then the coordinates correspond to the intensity of these different markers. Now, these markers are chosen in a way that you can distinguish different types of cells. So, for example, you can identify different B cells, or you can identify T cells in those samples. Now, what happens to this type of data routinely is that doctors will try to use it to identify cancerous subpopulations. And what they do in practice is they trace out the shape of these data sets in 2D projections. So they select pairwise markers, they project the data to these 2D versions of the data, and then they trace out this shape by hand, and among other things, they assess empty spaces. Now, this is done manually and is, of course, time-consuming, subjective, and therefore also prone to errors. And with a TDA hat on, if you hear about these empty spaces, then it appears like a natural application of TDA in particular, because then you don't have to look at these 2D projections, but you can take the full high-dimensional point cloud into account. So the goal here in this project was to take data which is taken at diagnosis and then objectively predict the risk of relapse from this data. Why is this important? Because then you can start tailoring treatment plans based on the individual risk. So, for example, if someone is at high risk of relapsing, you can skip aggressive or toxic chemotherapy, which will likely not be very effective. And you go on to use more aggressive chemotherapy or expensive CAR T cell therapy as a first-line treatment in the hope that this person will then not relapse. Now, what we wanted to do is use TDA in combination with machine learning to characterize shape in these point clouds. And in particular, we were interested to look at which of these markers were particularly relevant for this shape-based classification, and develop pipelines which could be translated into practical clinical pipelines down the road. So we were incredibly fortunate to be able to work with a quite exceptional data set. So this was a flow cytometry data set which included 16 different markers, including B-cell markers CD10 and CD20. This is important because we're looking at a B-cell subtype of this disease, and differentiation maturation markers CD45 and CD38. And what was exceptional about this data set was that we knew the treatment outcome over time. So we knew that 80 of these patients had not relapsed and 16 had relapsed. And this is actually, so you might think that this is a very small data set, but these types of data sets are quite rare. And given the context, this is a very good data set to be working with. And we were very fortunate because Salvador, who we were collaborating with, his PhD was funded by parents of affected children who put together money for a PhD project and gave their children's data to the project to be studied. And so this is really an exceptional opportunity to be able to work with this kind of data. So the pipeline that we were using was we had these patient samples of bone marrow, and the doctors then gave us the flow cytometry files of these. They pre-processed these by selecting the B lymphocytes, so the cells that were most relevant to the task, and then gave us this to find shape biomarkers that were relevant for predicting risk of relapse. What we then did is we applied Vittorius Rips to these point clouds and focused mainly on dimension 0 and dimension 1 barcodes, and in some cases then also on persistence images for our results. So the first thing that we wanted to do, we had this lovely 16-dimensional point cloud, and we wanted to stick it directly into Vittorius Rips to not go via 2D projections, which is done in the standard analysis of these data sets. And then we quickly realized that these data sets were far too big for a full Vittorius Rips computation. And so we decided that in a first step, we would go in line with standard clinical practice and just consider 2D projections and run Vittorius Rips on these to just get simple barcode descriptors and see if we were even able to pick up anything in these 2D projections. So for this, we ran Vittorius Rips in dimensions 0 and 1, and we then constructed 8-dimensional summary vectors of the barcodes, including the maximum persistence, minimum persistence, mean persistence, and standard deviation. And we did this for all pairwise combinations of the markers. And we then put this into a random forest classification to classify between patients that had relapsed and patients that had not relapsed. The advantage of this was that we could identify markers with the most information content using these random forests. And what was amazing for us was that with these simple summary statistics, we were able to confirm the importance of known biomarkers CD10 and CD20, which are the B-cell markers, and CD45 and CD38 in a completely independent manner. And so we are very happy with this first step, and we decided to retain these 4 markers for our subsequent analysis. And we then looked again in the barcodes of the pairwise combinations, which specific topological features were particularly relevant for distinguishing relapsing from non-relapsing patients. And the second thing that we did is what we refer to as persistence threshold curves in the paper. I think it has many other names as well in the literature, which is where we look at the number of features whose persistence is above a set of thresholds, and we change the threshold and then get a curve that describes the persistence in these barcodes. So we did this, and we found that the mean persistence threshold curves have higher values in relapsing patients than non-relapsing. So we have more persistent connected components and loops in relapsing patients, except for one parameter combination, which is actually consistent with the medical hypothesis that in relapsing patients, you have more heterogeneous cell populations. So if you think about a point cloud, which is more heterogeneous, has more sort of spread out points, then this will also give rise to larger loops or more disconnected connected components, which are then found by Vittorius Rips. Now, for dimension 0, what is often done by clinicians is they use a type of clustering. So we were able to reproduce these results, which are typically done on the full data set for these selected dimensions. What was interesting is when we applied this to the full 4D point cloud, these persistence threshold curves did not help us to distinguish relapsing from non-relapsing patients. So we found that very interesting that in the 2D case, you kind of, the simple summary statistics such as these curves will help you distinguish, but not necessarily if you add in the full information from the 4D point clouds. Now, we still wanted to go into the full 4D analysis. This again required us to reduce the point clouds. So we chose 10 to the power 4 landmarks by the maximum algorithm for computations in dimensions 0 and 1. And then 10 to the power 3 for common calculations of Vittorius Rips in dimension 2. And we then decided to use persistence images as input into logistic regression and support vector machine. And this was where we were quite surprised. So we got really high accuracies, in particular for the dimension 0 and dimension 1 persistence images. Where we had 100% accuracy on this data set. Of course, this is a small data set, so one needs to be careful. But we were quite happy with this because if you do a random forest on clinical variables, so you look at, for example, mutations, age, and variables like that, so very simple summaries of these patients, then you only achieve 86% accuracy when predicting who is at risk of relapse. And if you look at the flow cytometry data with the method that the clinicians use in the 2D projection, then they get 91% accuracy. So it seems that we're really capturing something in addition. Now, the data set was quite unbalanced. We had only 16 patients that relapsed and 80 that didn't. So if we account for that, we still have these very high accuracies. Now, if we look at the coefficients of the logistic regression decision functions, these can point us to areas in the persistence images that are particularly relevant for distinguishing between these two subtypes of patients. And we found that in dimension 0 and 1, we had sort of hotspots where the important distinguishing features in the persistence images lie, whereas in dimension 2, we don't necessarily have these strong hotspots. And this was probably also reflected in the fact that dimension 2 wasn't quite as good in the prediction of whether someone would relapse or not. So based on this, we came up with 3 different pipelines, depending on the level of sophistication that one would like to use in practice. The first one was just based on these 2D projections and extracting from the barcodes the most important distinguishing features between relapsing and non-relapsing patients. And so, for example, here in CD10 and CD20, small connected components and large loops point to relapsing patients, while we don't have this in non-relapsing patients. CD10 and CD38, as well as CD20 and CD38 have large loops in their barcodes, which we don't see in the non-relapsing patients, where in the second case, we see more small loops. And so here we were really able to distill out very concrete features to look for in these point cloud projections. As a second level, we would recommend using persistent homology on the 2D projections and then constructing these PT curves, so these persistent threshold curves, where we had a statistically significant difference between the relapsing and non-relapsing patients. And then finally, if one has access to the full computational tools that we use, so persistence images and machine learning, then using the 4D point cloud and persistence images to feed into support vector machine or logistic regression gives us at least very high accuracy. So TDA in combination with machine learning really significantly improves the risk prediction, at least on this small data set, using data at diagnosis. So not at the time when these patients relapse, but really way before then. And what we also saw was that we, in particular, were able to improve this for patients that were previously falsely classified as being low to intermediate risk, but later relapsed. And this is actually a very high part of the relapsing patients. So I think it was about 87% of the relapsing patients had previously been told that they had low to intermediate risk. So this really makes a huge difference. What was interesting from the TDA side was that we found that simple barcode descriptors perform better on 2D data compared to 4D, but then taking the full information into account using persistence images in 4D did even better. And what was also nice was that the cellular heterogeneity hypothesis for relapse patient was consistent with what we found in the topology. But again, this is a small data set, so there is some risk of overfitting, and we'd be very keen to look at this in a larger data set, or also in different hematological malignancies, because in many of those, this type of data is really routinely taken. Okay, so we know clearly how to deal with point clouds when we have points all of the same type, and we're interested in their shape. But what happens if we have a point cloud where the points are of different types? So for example, different cell types. And we don't just want to run Vittorius rips on the individual cell types, but we really want a method to encode relationships between these different cell types. Now, why is this relevant? So there's very many areas, application areas, ranging from biology to geography, as well as ecology, where this type of data is routinely collected, and there's a great interest in understanding the interactions between different species. In biology, this is motivated by methods coming out of spatial biology. So this is a whole range of novel experimental techniques which can spatially resolve, for example, transcriptomic data or gene expression data, different proteomic data, and so on. Now, one of the goals that was kind of announced when these methods first started coming out was that we would finally be able to precisely characterize the tumor microenvironment. So this is a kind of ecosystem around the tumor where the tumor interacts with its host. And this consists of, for example, blood vessels of the host. There can be immune cells in there. There can be different types of cells of the host in this vicinity of the tumor. There's lots of different chemicals that can lead the different cells to interact in different ways. So there's a lot happening in this microenvironment, and it's thought to be incredibly relevant for disease progression and also response to treatment. So being able to precisely characterize this tumor microenvironment and the cellular interactions therein, as well as the local architecture, is of great interest in oncology. So we have all these beautiful techniques, but are we close to this? And the answer is actually no, because there's a lack of methods that can fully exploit the available information that is contained in these types of data. So what is very often done is that people start counting cells and then distances to nearby cells of another type, but you completely lose the spatial resolution in this case. Slightly more sophisticated approaches are using machine learning, which can take spatial information into account, but there you lose interpretability. At least this is very often the case, although there's been a lot of advances in this area. So again, from a topology perspective, it looks like topology can really bring something to the table here, because we can capture spatial information across multiple scales, and we don't lose interpretability. So it seems like a natural kind of application if we manage to encode these spatial relationships. So one example of such spatial data are multiplex immunohistochemistry images, which are very common in cancer research at the moment. So these show the spatial distribution and intensity of up to 40 biomarkers in 2D biological tissue. And you have a variation of biomarker intensities here. What this allows you to do is that you can distinguish lots of different cell types. So for example, tumor cells, muscle cells, macrophages, T cells, and even subtypes of the same type of cell. So for example, different macrophage subtypes you can distinguish with these markers. And so this allows you to study the spatial and phenotypic heterogeneity in such tissue, given the right methods. Now, if we want to develop such methods, it might not be the easiest to start with actual biological data. Anyone that has worked with data from biology knows that it can be very messy, especially if you don't know the technique that you're applying very well yet. So what we turned to when exploring our ideas was synthetic data coming from an agent-based model. So this is a mathematical model that was developed by Joshua Bull and Helen Byrne at Oxford. And it was motivated by the observation that in vivo, the ratio of different macrophage subtypes can be relevant for prognosis. And so what they set out to do was to simulate interactions between macrophages and tumor cells in the tumor microenvironment. The output of this model are the spatial positions of 5 different cell types. So we have tumor cells here in red. Then there are necrotic cells. So these are cells that are dead, usually within the tumor. There are stromal cells, which are the blue cells kind of surrounding of the other tissue around this tumor. Then there are blood vessels around the tumor and macrophages of dynamic subtypes. So these are immune cells that can either be working against the tumor or for the tumor, but we'll see more details about this in a bit. Now, related to these macrophages, there are 2 parameters that can be varied in this model, and depending on what combination you choose over time, you end up in a different qualitative regime. So here in this yellow area, we have an equilibrium between the tumor cells and the immune cells. So we have the tumor here in red sitting in the middle, and then the yellow cells around it are the macrophages. If we choose the parameters slightly differently, we are in an elimination regime. So the macrophages have completely destroyed the tumor and are getting on with their life after that. Then in this red regime, we have escape of the tumor. So this means the tumor starts going towards the blood vessels, which can then lead to metastasis down the line, so spread across the blood vessels into different parts of the body. So how does this model work? So initially you have blood vessels, which produce macrophages at a set rate, which is related to this parameter C1/2. Which is in response to a chemical that is being produced by the tumor cells. These macrophages also have a certain sensitivity towards this chemical, which is given by the parameter chi, which can also be varied. And then once they come out of the blood vessels, they start to migrating towards tumor cell clusters. And once they are there, they might kill a tumor cell, turning it into a necrotic cell. Now, while these macrophages are close to tumor cells, they're exposed to a second chemical that the tumor is producing, which is TGF-beta, which can drive these macrophages to go from an anti-tumor behavior to a pro-tumor behavior. So pro-tumor macrophages produce another chemical, EGF, which can then attract tumor cells, and at the same time, they are attracted back towards the blood vessels. And so what happens is that they migrate towards blood vessels, pulling tumor cells behind them. And so this is how you then end up with an escape behavior of the tumor, depending on the parameters where the tumor can go towards the blood vessels. So the 2 questions that we wanted to answer when looking at simulations from this model were the first one, if we get an image and we don't know if we're dealing with pro or anti-tumor macrophages, can we tell just by the spatial distribution of the different cell types whether a particular image is predominantly consisting of pro-tumor or anti-tumor macrophages? In the second case, we wanted to be able to take an image from a particular parameter combination and then tell whether it is coming from an elimination, equilibrium, or escape regime. So the 2 tools from topology that we used here was the first one, the Dowker complex. So this focuses on pairwise relations, We vectorized this using persistence images and then used this to gain insight into whether or not a particular image was predominantly M1 or M2, so pro-tumor or anti-tumor macrophages. The second method that we used was looking at multiple relations at once, so not just pairwise relations, and is based on the witness complex, where we construct different filtrations per cell type that we have in these images, and then construct topological distance vectors, which we can then cluster. So I'm going to go into more details of the methods now. So the Dowker complex was introduced by Dowker in 1952. And basically, if you have two distinct point clouds, U and V, which in our case can correspond to tumor cells and macrophages, for example, and some relation on these 2 point clouds, then the Dowker complex is a simplicial complex that represents this relation. You can build a filtration on this if you do a Dowker complex at parameter epsilon, which is a simplicial complex that has, for example, U as the potential vertex set, and then includes the n simplex based on vertices in U, if there exists a V in the other point cloud, such that the distance of the UI and V are small or equal to epsilon for all the vertices of the simplex. And then you can vary the epsilon to create a filtration. So to give you an example, here we have a point cloud U and V. And then if we choose as potential vertices U, then we start blowing up little epsilon spheres around our vertices in U, similar to the Vittorius-Ribs complex. But now we just put an edge between 2 vertices if we have one point of the other point cloud in the intersection. And so this is how we build our simplicial complex at parameter epsilon. Now we can, of course, also do this on the other vertex set, where we can see that in a way the simplicial complex highlights the shared features, and it doesn't build edges on features that are not shared. Now, the nice thing is that Dowker showed that it doesn't matter if you choose U or V, you're going to get the same homology on the simplicial complex, and this was later extended for Dowker filtrations. You're going to get the same persistence diagram for both choosing vertices in U and vertices in V. So we do this now to our images created from the agent-based model, and we do this for macrophages versus tumor cells versus blood vessels, and macrophages versus blood vessels. We produce the persistence diagrams, then construct persistence images, and then feed these into a support vector machine to predict the dominant phenotype in a particular image. To show you some examples of these persistence diagrams and what they capture. So we have 3 different images here, and then the 3 different combinations of cell types over here. And so, for example, here in the macrophages versus blood vessels, we have one persistent feature showing up in dimension 1, and this corresponds to this shared loop between the macrophages and the blood vessels in this particular image. We don't have this in the other 2 images. We have a maybe slightly persistent loop here, but it's not as persistent as before. And similarly, if we look at the connected components, we have one persistent connected component in the tumor cells versus blood vessels in the middle image. And this corresponds to sort of shared connected components as the tumor starts already to migrate to these blood vessels. So let's see how we did on the support vector machine and the prediction task. So in red here, we have non-topological features being considered for the prediction. So this means we're taking into account the number of cells for each cell type, and then the distances, average distances to the other cell types. And so this achieves below 80% accuracy on this task. And then in gray, we have the Dowker features, which do much better already, not in all cases, but in many cases. And if we combine them in particular, then they do very well. And if you run Vittorius RIPS separately on the different cell types, then this also performs better than the standard features, but it doesn't do as well as the good performing Dowker features. And even if you concatenate all the different Vittorius RIPS filtrations into one vector, you will not get the same accuracy as when taking the relations into account in the Dowker complex. So we thought that was very encouraging, that being able to encode these relations really leads to improved accuracies in this particular task. We then also did some multi-dimensional scaling where we compared the Vittorius RIPS features to Dowker features. So this is, you can think of as a version of PCA. And here you can see that the Vittorius RIPS features don't distinguish between the M1 and M2 dominant images as well as the Dowker features do. So let's come to the second method. This is the multi-species witness filtration. This is inspired by the lazy witness filtration that was introduced by De Silva and Carlson in 2004. And the lazy witness filtration basically selects a set of landmark points in a larger point cloud and then constructs simplicial complexes with vertices on those landmarks. And the remaining simplices are then defined depending on the existence and vicinity of witness points in the point cloud without the landmarks. The idea being that this kind of gives an intrinsic Delaunay triangulation of this point cloud and also reduces the computational cost for very large point clouds. So we now build on this and introduce the multi-species witness filtration. So here we have a point cloud that consists of the union of separate point clouds, and we fix one of these point clouds as being the landmarks. And then we construct a Delaunay triangulation directly on these landmarks. Now, the remaining cell types are used as witnesses for the simplices in the Delaunay triangulation. And we now filter by the number of witnesses for each simplex rather than the distance to the witnesses. So for a point to be a witness point for a particular n simplex, we need the vertices in this m simplex to be the closest landmarks to this witness point. So each vertex is going to have as witnesses those points whose closest landmark this is. And a point is a witness for an edge if the two vertices that make up that edge are the closest two landmarks to this particular point. For a triangle, we have the three closest landmarks that are needed for this point to be a witness for that triangle. Now, the multi-species witness filtration then filters the simplicial complex by bringing in those simplices with the most witnesses first, and then the second most, second, and so on. And we normalize this by the number of witnesses of a particular cell type to avoid that if we have some cells where there's a lot more of them than other cell types, then we won't distinguish that much between them as if we would if we were taking absolute numbers. And if we include a simplex, then we also make sure that we include all of its faces. So here is an example. We have a point cloud that consists of these purple points and then yellow points and these blue stars. So we choose our purple points as being our reference or landmark points. And on these, we construct a Delaunay triangulation. And then we can use, for example, the yellow points as witness points. And we include these simplices that have the most witnesses, so 2 in this case first, and then those with 1 witness, and then all the remaining ones. And we do the same thing for the blue stars. So we include those with 2 witnesses first, then those with 1 witness, and then finally we have the full simplicial complex. So the idea here is that we're filtering the same simplicial complex, but in different ways, and these different ways are informed by these different witness points. So now if we take distances between the persistence diagrams of these filtrations, these will really highlight different spatial distributions of the witness points. So in our example, what we do is we choose the blood vessels as being our reference point. So those are the ones that we build the Delaunay triangulation on. We then produce different filtrations for the tumor cells, necrotic cells, and macrophages of type M1 and macrophages of type M2. We do this for each image. So we produce 4 different filtrations for each image. We then take pairwise distances between the persistence diagrams. So we use the bottleneck distance and Wasserstein distances for dimensions 0 and 1. And so each image now is turned into a topological distance vector, and we then use k-means clustering to see if we can reproduce the 3 qualitative behaviors. So here are the results. On the left, you have experts annotation. So here, Joshua Bull looked at 1 simulation per parameter pair and estimated if this looked more like elimination, equilibrium, and escape. In the middle, we have the fully automated k-means clustering of our distance vectors using 3 different clusters. And we can see that these align really nicely with the expert annotation. The only differences are kind of in these transition regions between different qualitative behaviors. We did this over 20 simulations per parameter combination. And so we also looked at how often the majority cluster was voted as being the cluster in these 20 simulations. And you can see that there's a lot of consistency across the 20 simulations, again, within these qualitative regimes, but on the boundaries between them, it's more inconsistent. And this is consistent also if we ignore macrophage subtypes. So we don't look at M1 versus M2, but we just classify them as being macrophages. Now, what was really interesting is when we looked at 4 clusters, then these boundary regions were highlighted as their own cluster. And we found that really cool because it seems to be that there's really something that the multispecies witness filtration is picking up here that is different in these transition regions. We, however, only get this if we include macrophage subtypes. So we say that we have an anti-tumor or a pro-tumor macrophage. If you do the same thing on simple feature vectors, so again, looking at the number of cells of each cell type and then the distances to the other cell types on average, then you can't get you don't get very nice alignment of these qualitative regimes. You get some alignment, but not all of it. And in particular, if you use 4 clusters, you do not get these boundary regions that are highlighted. So we're really excited about this because we think it's an important step to really capture relationships in spatial data and can give rise to a whole new subfield within TDA, which is relational TDA or relational persistent homology. We can capture spatial relationships beyond single system methods. And in particular, in our application, we found that the Dowker features can predict dominant macrophage subtype, whereas witness features can capture qualitative behaviors of the model that we used. And we'd now really love to test this on real-world imaging data. So thank you very much for listening. And I'd like to thank all my co-authors and mentors, in particular, Salvador, Victor, and Helen on the Leukemia project, and then Iris, Jay, Josh, Helen, and Heather on the Relational Persistence project, and EPFL, L'Oréal, UNESCO, and EPSRC for funding my work on this. And thank you very much for listening, and I'm happy to take questions.
Okay, thank you, Bernadette. Perhaps we can all thank Bernadette by unmuting ourselves and clapping. Questions?
I have a question. So a very nice talk. Thank you very much. I was wondering, in the beginning, you showed this picture where the doctor was hand selecting the points. And it looked to me like the doctor selecting points that are particularly dense, so dense regions. Whereas then the Viatoris persistent homology would consider every point fully, not just the dense points. So have you considered doing something like density RIPS, or maybe density RIPS would be a 2-parameter persistence module that's maybe a bit not so handy to work with, but maybe a slice of density ribs or degree ribs, or something in that direction where you just look at the dense regions?
So we haven't done that so far. So the density, I think in a way they're trying to assess the point cloud shape. And so it makes sense to kind of go around the dense regions. It's not clear that it's necessarily the density that they're looking for, because they're also assessing the empty spaces in between, right? And those also depend on, in particular, not dense parts. But it's certainly an option to look into that. So far, we've just been really happy to have really high accuracy taking TDA in comparison to those hand-drawn features. But yeah, that would certainly be an option.
Yeah, your results are already great without doing that. It's just like maybe they would be able to improve even more or so. Yeah, it's really great what you presented. Thank you.
More questions for Bernadette?
Question.
Yeah.
If I may. Thanks. Teresa, you were a bit quicker than me. So I have two questions actually. My first question is, in this last method that you're working on, the witness method, you're taking the vessels, right, as the sort of the first set that determines the triangulation, and that gives you certain results. What would happen, or does it make sense to also like twist it and take one of the other three groups as the reference points? Like, would you be able to capture other information?
So that's something we want to look into. So the reason for the vessels is that they're quite nicely spread around all the other cells. And so that's why we chose them as a first step. But of course, it'll depend on how those reference points are spread out in the image. So for example, if you chose something that is just located in the upper left corner, then that will not be very helpful. So something that I'm investigating at the moment is if we just use an artificial grid kind of in the background, does that give better results actually than choosing one of the cell types as a reference?
But then you would have sort of three different, because, well, from what I can see, you have three different cell types, right, that you consider. And if you increase the number of types, like how bad does it get? How quickly does it get bad, I guess?
So we haven't pushed it to the limits yet. I mean, our data sets are thankfully quite small in this artificial setting. It'll be a whole different picture in real data. And there, I think we'll have to think about the computational feasibility.
Okay. Thank you. I have the other question, maybe we can also leave it for later, is what do you have on this thank you slide? But
Yes, so this is from a different project where we looked at local persistent homology to detect geometric anomalies in data. So it kind of flags up. So this is the conformation space of cyclooctane projected down from R24 to R3. And you can see the red points are close to intersections of the sphere and the Klein bottle in this data set.
very, very cool.
Yes, sadly,
Thank
there
you
wasn't
very
time
much.
to also talk about this.
More questions for Bernadette?
Well, maybe I can ask a quick question. So first of all, thank you very much for the great talk. I think in the beginning you said that I was in the first example that sadly the two-dimensional persistent features were not quite as good in predicting the outcome as the zero-dimensional and one-dimensional features. But if I remember correctly, you also scaled down the point clouds for the two-dimensional features from I think 10 to the power 4 to 10 to the power 3. Have you like run the same experiments with the zero and one-dimensional features also on these like scaled down point clouds and checked how well they perform there?
No, so we didn't do that. We wanted to get as many points into the computation as possible, and so we only reduced for dimension 2. But yeah, it's definitely possible that the problem is that we're just reducing the point cloud so much that there's not much information in dimension 2 anymore.
I mean, yeah, I'm just curious, but thank you for the answer.
Okay, more questions for Bernadette?
If I can A bit more technical question just in this direction where you were reducing the size of the point cloud, right? You are computing Vietore strips of dimension 0, 1, and 2, right? I did this in a project for 10,000 points in dimension 300. And it was kind of feasible. It took, I think, like 2 days, and I needed like 100 gigabytes. But then, if you are using cluster, it's viable. So, what was the problem? Do you have even more than 10,000, or were there not the computational resources, or?
So it was 10 to the power of 5 points, and these computations we did in 2020. So it's entirely possible that nowadays it would be much more feasible.
Right. And maybe one more question to the projections. So with these silly statistics that worked well on 2D projections, but not in 4D, if I understood well, you projected in a way where you just take 2 markers and ignore the others, right? Did you try some different orthogonal projections to some different planes, be it an arbitrary plane that somehow cuts through your data, or something like PCA plane dimension 2?
So we didn't, and the reason for this was that we wanted to stay close to the what the clinicians were doing. So we felt like if we're using projections, then we should use the ones that they're using in practice already. But this would definitely also be an option to explore.
Do you have some intuition there whether that could improve, or no idea?
So I mean, so we reduced it down to the 4 important biomarkers, and I think on this 4D point cloud, it could possibly help to project it down in a more sophisticated way. I'm not sure about the 16-dimensional point cloud, which was the original one, if that would give anything that would be as interpretable.
Right, makes sense. Thank you.
Okay, so thank you, Bernadette. I'm going to stop the recording now, but we can take the rest of the questions offline. Thanks again.